Though neurons are the cell you probably think of first when talking about the brain, did you know they have a host of bodyguards keeping them safe? Glia are cells that provide support and protection for neurons and are a vital part of keeping your body functioning properly. As you can imagine, cells with such a big responsibility can be destructive if they become mutated. For example, glioblastoma, the most common type of brain cancer, is super aggressive. Many patients are given a life expectancy of 1 or 2 years after diagnosis, and that's with the help of treatment.
In many cases, we recommend either surgery to take out tumor, as much of it as possible, or a biopsy to establish a diagnosis. After surgery, patients will typically undergo standard therapies, that is, a combination of chemotherapy, a drug called temozolomide, and radiation. And that's been our standard treatment for almost 20 years now. And despite these treatments being around for so long, we have not really identified better treatments for these tumors.
That's Dr. Wajd Al-Halou, a neurosurgeon and assistant professor in the Department of Neurosurgery at the University of Michigan. Despite being able to remove much of the tumor in some cases, Al-Halou says it's never enough to outrun the cancer's rapid growth.
Sometimes what we can achieve something called a gross total resection, meaning we have removed everything that is visible on the MRI. But the reality is, even if we achieve this extensive resection, the tumor is actually beyond that. It has traveled centimeters into the brain. And so even though surgery has shown to help improve prognosis, survival, and quality of life, etc., it does not cure the tumors.
Glioblastomas are so hard to treat partly because no two tumors are the same. There are significant genetic differences, which makes it tough to develop a drug that would be effective for multiple patients. However, the one factor that these tumors have in common is that they all need to consume energy in order to survive and spread. And though there's been a lot of research around cancer, Al-Halou says the one area that's been overlooked is what these cancerous cells use and how they metabolize energy. Al-Halou and a team of researchers sought out to uncover these mysteries with their new paper published in the journal Nature. They had guessed that glucose was a good place to start their search since all living cells use it.
It's a common source of carbon, is how our cells run most of their metabolic processes. But the reality is, like, one cell in the body may use glucose to do one thing, so your brain may use glucose to make neurotransmitters. Your liver may make enzymes, but we don't know what the cancer cell is using it for. So if we know what that cancer cell is using that glucose for, then we can attack that pathway. We can block that pathway.
This research project is unique in 2 ways. The first is that it's a deep dive into the metabolic processes of tumors, and second is that it brought together various teams of researchers to contribute their expertise.
Glioma metabolism has been studied in the past, but not in the way that we have done it as a group. I think what we've realized in the brain tumor world is that the old-fashioned model of one scientist discovering things on their own just doesn't really work. You need people who have significant knowledge and expertise in every specialty to make things work. So you need metabolomics experts, you need bioinformatics and engineering experts, you need neurosurgeons, and you need radiation oncologists. Without that, you'll never really get to the real truth about these tumors. And importantly, you won't be able to impact patients' lives.
The group used 2 methods in their study. They started with isotope tracing, which is a way to follow a certain nutrient, in this case glucose, through parts of the body. Al-Halou says that while this is a common technique, it hasn't been widely utilized in glioblastoma research. The second advancement the team was able to utilize was seeing how the tumors acted in human patients.
And most of the time when people study glioma, we're often studying it in a cell line in a dish or in a mouse model. And these are important ways for us to study disease, especially brain tumors. But to really know the truth about how a brain tumor behaves in a patient, in a human being, we need to study it in patients. And that's really the major leap that we made, is that we were able to study it in patients, and we're able to compare the tumor to the brain of that patient. So that's what was really unique. And that gave us the opportunity to make these amazing observations and discoveries that I'd never seen before.
All of their experiments were first done in mouse models, then they were able to give human patients glucose during resection surgeries and send a sample of the removed tumor to their lab for inspection. Their research revealed an exciting finding of how the cancer cells are obtaining and using glucose.
The cancer cell's priority is to grow really fast, divide, and invade into the brain. So it has to do a lot with very limited resources. So it shuttles a lot of its glucose to making these purines and pyrimidines to help the tumors grow. They also need other things to maintain their cellular processes and to survive. And so they end up, instead of making them, they end up essentially like stealing them from the environment. So it ends up using less energy. You don't have to make it from scratch. You essentially borrow it for somebody else.
Meaning that the cancer cells are stealing the glucose from the other healthy cells in the brain that need it to carry out their functions. Al-Halou and the team found that the tumor uses the sugar to create an amino acid called serine, which fuels its rapid growth.
That was an important discovery in our patient tumor samples. And then the follow-up question was, now that we've discovered that these tumors require specific amino acids but don't make it, can we starve them? Can we starve these tumors of this essential nutrient?
The second part of that question is, even if you are able to starve the cancer, will it matter? Will it impact the tumor's function?
And the answer was yes. At least in our animal models, the answer was, if you starve these mouse models of serine specifically, the tumors don't grow as fast. So how do we restrict serine? That's a difficult question. It's not something that you can easily go to the grocery store and not buy the product that has serine. Serine, unfortunately, is in lots of things. So it's not easy to do that on your own.
The group is now collaborating with a company that's manufacturing specific dietary products that don't have any serine in them. Another option the team's looking at is developing a drug that can block the serine from ever getting to the cancer cell.
Restricting diet in patients is challenging because you're talking about a patient who has a very bad tumor. You're asking them to completely change their lifestyle and their diet to help prevent the tumor from growing. And I'm sure a lot of patients want to do it, but it's not easy to do. It's something that we worry may be hard to adhere to in every patient. But if we can find better ways to block the use of serine in the cells or the transport of serine to the cells, it may actually be more effective.
Which gives the team a lot more work to do moving forward.
Even though this is a huge, huge accomplishment to find how these brain tumors consume energy, what they use glucose for, that serine is a potential vulnerable target, it's not the end. The next step is the easy step. Can we restrict serine and glycine? Can we block serine and glycine? Our work continues. We're trying to find other amino acids, other nutrients that these tumors need. So these studies continue. We're trying to find the next thing. And the ultimate goal is we are not going to rest until we find something that is effective in these patients.
You can find more information about Dr. Wajd Al-Halou and all of our guests on our website, ingoodhealthpodcast.org. For more behind the scenes, follow In Good Health on Facebook, Instagram, and X. Our writer-producer is Kristen Farah. Our production manager is Jason Dickey. I'm Greg Johnson.